All right, so basically the first slide, as you see, says something. It says a long journey. No, oh, sorry. I'm sorry, I forget which button is which button now. A long journey starts with a single step. This step is 10 to the minus 15th of a second, a femtosecond. And this step then leads to all the food and oxygen. So that from a fast reaction of femtosecond to the minus 15th of a second, we have food and oxygen. And that's something to imagine and something to remember. Okay, so what else is in the slide? Well, there's a tea, there's a truck. So the plant is making the light photosynthesis, and this diagram was made by an artist named John Hooper. And so I'm showing you that all these activities are based on what happened here. So, as I said, major advances in understanding photosynthesis and biological systems in general occurred when new knowledge and techniques from other sciences, physics, chemistry, were applied. So today the biology depends heavily on what the physicists did for us and the chemists did. And as you will see in my lecture, you will see that almost all the Nobel Prizes in photosynthesis have been given in chemistry. So therefore, biology cannot ever separate itself, and I'm very glad that in this institute, the wholeness of science is being understood and perpetuated. So my book, a book by, I keep forgetting, edited together with Tom Beatty, our guest, who is no more, and myself, Discoveries in Photosynthesis, and it has chapters written by the discoverers themselves. All right, so what I'm going to do today is to talk about the steps that lead from the time exciton or photon comes to the time primary charge separation takes place. And to remember what a femtosecond is, I show you that there are as many femtoseconds in a second as there are seconds in 31.7 million years. There are some typographical English mistakes, so please forgive me for writing at 2 o'clock at night in Urbana, and I haven't corrected the slide yet. All right, so let us see. We want to remind you, one of my interests is to make sure that I say the same thing several times so that you will not forget it. So, reminder is, it is a conversion of light energy into chemical energy. It is the source of all our food and most of our energy resources on Earth, and its exploitation is necessary for sustainable humanity. And again, I enjoyed your brochure, which uses the word sustainable humanity. That's where we are all after. That's why photosynthesis is a very important and crucial area to be had and to be go forward with it. And this is the diagram. This is the artist's diagram showing a leaf of sunlight conversion into energy. It is done in Arizona State University and it now hangs in the walls of Professor Blankenship in Washington State University. All right. Now, to give you a feeling of the whole process, again, I must say that many of my slides are taken from friends who are in the field and made it as my slide, but I always give the credit. This slide was shown to me, this idea, by Professor Schultz in University of Toronto, Canada. And of course, he got it from somewhere. About 100 billion tons of biomass is produced by photosynthetic organisms per year. This means that photosynthesis is producing biomass equal to 2 Egyptian pyramids per hour. So it's a very huge process, but it's not enough. It's not enough. So it is amazing that we're still throwing away a great deal of sunlight. We need to harness it by both natural and artificial means. And that's the reason why we are interested. And I mentioned to you that the field is here because it's not only biology, but physics and chemistry has been in it. And most of the Nobel Prizes, so I'll show you two slides of the past Nobel laureates, and you'll see what they have done. Very briefly, the top slide, Richard Willstatter, oh sorry, excuse me, I'm walking around. Richard Willstatter for the chemistry of chlorophyll. James Franck for many of the ideas of physics, some of it we will discuss today, mention today. Hans Fischer, the second one for chemistry of chlorophyll, 1930. Otto Warburg for the physiology, in physiology or medicine, not in chemistry, for discovery of most of the enzymes that you have in respiration. Paul Kara and Richard Kuhn for the structure of carotenoids, the yellow pigments that we are using. Severo Ochoa for pyridine nucleotide. Melvin Kalman, we heard in the first slide, received the Nobel Prize for work he did with Andy Benson, or I should say Andy Benson worked with Calvin, and James Basham we mentioned, and Robert Woodward for chemistry of chlorophyll, again for synthesizing chlorophyll in the laboratory. So these are the next group of Nobel laureates, are some that I know personally. I have talked with them. George Porter, for flash photolysis. He was the first person to challenge me on my primary photochemistry work that I did with Mike Wasilewski, saying that I was wrong, but it turns out I was right, and he was wrong. And that is another beauty about top scientists like Porter, that yes, when they see something that was missed, they agree that yes. Now it is not a matter of wrong or right really, it's a matter of interpretation for the time, and he was a wonderful person who I knew. The second, James Norris was his professor. The third, Peter Mitchell. Mitchell's theory, you have a membrane, protons move from the outside to the inside, you have proton gradient and a membrane potential, and it goes through the membrane again through the enzyme ATP synthase, you make ATP. And that is the Mitchell theory, and it has been proven by basically two very important people whose pictures I don't show you. One of them is Wolfgang Junge in Germany, and another is Andre Jagodorf in USA. Those two people, the work of those people proves beyond doubt that the Mitchell theory works in photosynthetic systems. Then the third group, the second line is Hartmut Michel, one is Eisenhower and Huber for the crystal structure of bacterial reaction center. And I don't know personally Huber, but I know Hartmut Michel. He visited my laboratory, and I met Eisenhower. The last set is Rudolf Marcus for the theory how electrons move in chemistry and in biology. And it's known as the Marcus theory. I will not describe it to you, but one thing, I have a personal attachment to Professor Marcus, because when I was teaching a course in photosynthesis, about 40 lectures I gave, and Professor Marcus came to my class every day, and he was there 5 minutes before the students came, and he did 15 minutes after the students left, because he was very much interested in knowing whether this theory would apply to photosynthesis or not, and it of course did. So he remembers, and he sometimes jokes with me. He said, "Oh, he's my professor." Of course, that's not true. He was a very senior professor himself, and that's profound. And then Paul Boyer and John Walker, they received the Nobel Prize. In my next lecture, I will talk about their research, which they said how ATP is synthesized, what is the mechanism of the synthesis of ATP. Okay, so with that, let me move. So now, again, to remind you, that although the details differ in various types of organisms, the basic chemical mechanism is essentially the same in all organisms that do chlorophyll-based photosynthesis. There is light harvesting followed by charge separation. That's the title topic. That is the topic of my talk today. So I hope I get to it. But before I get to it, you have to recognize this is Carl Booth at the University of Illinois at Urbana. He just passed away recently, and this is the scheme of evolution that he made. And as you can see, that he has three forms of life. He discovered that there are two types of bacteria. One bacteria, the other archaebacteria, or we call archaea, and third eukaryote. So the cyanobacteria are in the bacterial line lineage, and then they somehow are responsible for the plants and plantae and algae. And so these are the only two oxygenic photosynthetic systems, which is what I am talking about. I'm not discussing the other bacteria. So in evolution, we have to recognize an oxygenic photosynthesis. According to most people, came first, then the oxygenic, and then the aerobic respiration came. And this is the fossil cyanobacteria picture. So basically, we can see the cyanobacteria are the oldest, the photosynthetic bacteria may be older, the photosynthetic eukaryotes, and then of course the animals. Okay? So now, the last one on evolution is there was extensive gene recruitment, anaxygenic photosynthesis. This slide is taken from Professor Blankenship. By the way, his book is coming out on photosynthesis, molecular mechanism of photosynthesis. It's already finished in the revised edition. And in the between, there's transitional forms, Cleobacter, Chlorophyll, and then Cyanobacteria, then the plants. So these are very basic things that we cannot forget. So now, coming to the topic, finally, there is a division of labor among the light-absorbing pigments. The majority just serve to harvest solar energy and transfer that energy to a specialized few where that energy is fixed by doing chemistry. The majority form light harvesting complexes, and the special few from reaction centers. Now, how did we know about this? Was there a history behind it? Well, before I go to the history, so we call these absorbing molecules antenna molecules. Okay. Now, are all antennas the same? No. It turns out all antennas, most antennas are different. And there are very, very types of antenna. And the evolution of antenna had taken place many times, not once. And there are tremendous differences in the genome of the different antenna. So I just want you to point out that the antenna, excuse me, I keep forgetting the button. Just as there are different types and sizes of antenna to catch radio waves, photosynthetic organisms have different types and sizes of antenna to capture light energy. The function, ultimate function, is the same in all systems. So these are the different types of antenna that are capturing light energy. You got prochlorophylls. This slide is taken from a review. This part is from PACE and Science, and this is in Brand. So prochlorophylls, cyanobacteria, green sulfur bacteria, green, and we'll not talk about those, but they also have antenna. They also have reaction centers. We are focusing on plants and cyanobacteria. So that's the evolutionary tree, basically taken from Carl Woese and amended by PACE, who had worked with Woese. Okay, so now Nathan and Dunford have put another scheme for showing the different types of antenna. And you can see here the red algae have different antenna, the glaucophytes have different antenna, and plants have different antenna. But basically, there are many differences. The light harvesting complex is like this. The early light inducible protein is like that. And one helix protein is like that. Cyanobacteria here. Stress enhancement protein is like that. And so basically, there are tremendous variation in antenna. But the mechanism of energy transfer is basically very similar. So let's go back to this old scheme that I may have shown you the other day. So they have the so-called light and the so-called dark reaction. Because there's only one light reaction. That is the light absorption and conversion of that energy into chemical energy in the reaction center. There is no other light reactions. What we call light reactions, like water to NADP, these are not light reactions. Most of them are dark reactions. The same way in the Calvin-Benson cycle, which is supposed to be a dark reaction, the enzymes need to be activated by light, and therefore the use of the word dark reaction must be understood from that point of view. So, light absorption, let's say femtoseconds, excitation energy transfer takes place several picoseconds, that's 10 to the minus 12 seconds, and the primary charge separation, again in picosecond, 10 to the minus 12 seconds, electron transfer is a bottleneck, and it's a millisecond. And if you want to improve photosynthesis, really, I don't think you need to worry too much about the faster reactions. You need to worry about the slower reaction in the millisecond and in the carbon fixation. Okay, so you know what I said last time, water provides oxygen and CO2 speaks to carbohydrates. So this is a basic slide, teaching slide. Now, we all know where things are happening. Chloroplasts absorb light. Here's the chloroplast picture. Here's a picture from my Scientific American article many years ago showing the analogy of the chloroplast, the green chloroplast, and so on. But this is also. Now, light absorption, what the leaf does. The leaf absorbs almost all the light. You can see there's only a small window in green. That's why the leaves are green. However, as I said the other day, yesterday, that there's a lot of light here, energy is not absorbed. And Blankenship has written a paper in Science with many co-authors suggesting that if we were to take and improve plant systems for light absorption, one of the things we can do is to make photosystem 1 absorb that light, changes redox chemistry, and maybe make use most of that. I don't want to talk about chlorophyll fluorescence. Those of you who are interested, and I had a very good talk today about fluorescence, can meet with me. I'll be happy to talk about fluorescence. So now, one idea suggested that, well, can we make plants black? Is there good to make plants black? Well, you can absorb more green light. But as I said before, and I may say in the last lecture, Don Orton pointed out that if you are working with crop plants, you have to not consider a single chloroplast or a single leaf, but you have to consider the entire canopy of the plant. And therefore, when you do that, improving and making a plant black is not going to help because the lower leaves will not absorb any light and it will be wasted. So therefore, one has to think through what we need, what we want to do. All right, so that's the point I want to make. Shouldn't plants be black to capture the light? And I would say, I don't think it's going to help. So let us not try to do that, unless somebody wants to do it for the purpose of science. And here is the Cyanobacteria who really did it. Oops, sorry. Who really did it. Okay. And this Cyanobacteria is totally black. All right. Now, where does the process take place? Where the charge separation takes place is in the chlorophyll molecule itself. And this is, as I said before, Wistacher, Fisher, and Woodward got the Nobel Prize for. So what is the thing in chlorophyll? It is the ring. It is a single double bond in the chlorophyll structure which gives the property and redox property as well as the property of light absorption. These are carotenoids, and I've already mentioned the Nobel laureates for that. So now coming to the basics. Now, most of you who are in physics, this is the basic physics, and you are born. However, those of you who come from biology may not have seen this diagram, which is called the Jablonski diagram, after the Polish physicist. And before Jablonski, there was Perrin in France, so it's Perrin-Jablonski diagram. And what it is, it shows you what is happening. So if you put blue light on a plant, then you go from a lowest excited state of a molecule to the higher excited state of a molecule. And if you put red light on the plant, you go from the lowest excited state, which is called S0, to the S1 state. And once you have the photon is absorbed in femtoseconds, once you have reached this molecule excited state, you can see it basically has a lot more energy. It's like jumping. It's like full of energy. It has gotten the energy of the photon and converted it into, it's now an exciton, and it has all the energy that the photon had. So it's an excited state. So we call it, the physicists call it excited state. Oops, sorry, excuse me, I keep forgetting that. So this is the S1 state, and it's a singlet state, always. What does singlet mean? To those of you who have not know, two electrons spin in the opposite direction. When a photon of light comes, one of the two electrons goes to the higher state. And this is a singlet state. It is a singlet state because the physicists, the physical chemists will tell you that you put a magnetic field, you will have only single energy level, and that's a singlet state. But in chemistry, it's a 2 times J or 2 times S with a spin, electron spin is a plus half, a minus half. Plus half minus half is 0. 2 times 0 is 0. 0 plus 1 is 1. That's why it's singlet state. Most photosynthesis takes place from the singlet state. So these are the singlet state photochemistry. And then, now, all photosynthesis takes place from the red light, not from the blue light. Because when you excite in the blue light, all energy is lost as heat, and you come down to the S1 state, and this is from where you have photochemistry, photochemical trapping, fluorescence, heat loss, or energy transfer to another system, called further energy transfer. Okay, so we have to remember that when we talk about how plants protect themselves from excess light, we call it non-photochemical quenching, and one of the things that happens... Pardon me?
So, energy cannot go direct to KP.
No, because there is no energy. It is the lowest energy possible. It's only when the electron is in the excited state, when the molecule is in the excited state, it has the energy to do photochemistry. Why? Because photochemistry needs energy. The photochemical trapping or the charge separation needs energy. Why? Because the electron and the redox potential is such that without energy it cannot happen. So that's why it takes place from that level. Okay, any other question? No? All right. Now, the next principle. I showed you a picture of James Franck, the Nobel laureate of 1926 experimental physics. He gave this theory. How the molecule... So I already answered the question, what is the single state? Okay. So here is James Franck. He left Germany because Hitler was against the Jews, and most of the scientists who were at the top were Jewish scientists, and many of them had to leave Germany, they came to America and other countries. James Franck was one of them. My professor, Eugene Rabinovich, was another. So James Franck came to Chicago, and these ideas were already presented by him. So you have a molecule in the lower S0 state, then the first singlet state I showed you is the Jablonski diagram, and S2 state. So when an electron goes from here to here, and it vibrates, and it comes down to this state, and from there you have fluorescence, chemistry, and this diagram, this is called the Franck-Condon Principle. What does it mean? What did Franck say to us? Again, as I said, you may not want to ask this question, you're afraid to ask this question because it's a basic question, but I give you the answer. And the answer is simple, that when electron, he said, is light and a nucleus is heavy. And therefore, when a photon comes, an electron jumps up, it is light, and the nucleus stay where they are. They don't move. So, the equilibrium state of the ground state is here, and when you go to the excited state, it is not at equilibrium, so now the nucleus moves and takes a new stable position, which is here. So that's the shift from this position, or the distance between the two nuclei moves farther to get a stable situation. And then from there, the reaction takes place. Now, we're coming back to the history of the discovery of the antenna and the reaction. The very first paper showing that you have a large body of molecules and a small enzyme was that done by Robert Emerson, who was my professor, first professor, and his student, William Arnold, who was an undergraduate student from physics who worked with Arnold. And in this paper of 1932, Emerson and Arnold discovered, and so what we call today a photosynthetic unit. He also discovered how many photons of light are needed to give you an oxygen, why 2 light reactions is necessary, and he also discovered what we call the enhancement effect, that there are 2 light reactions and 2 photosystems. And he is a key person to have made the major discoveries. He died long before he could be fully recognized. He was a member of the National Academy of Science, very young member, but never further. And he died in a plane crash on February 4, 1959, when I was his PhD student. Okay, next slide shows the results from 1932. It shows this experiment was not done in Urbana, it was done in Caltech. I realize that you have a connection with Caltech at Berkeley, and it was done at Caltech. And what they showed is they measured how much time is necessary. That 1 degree Celsius, you have the rate is slower than at higher temperature. But we will not discuss this. But what we will discuss is the following. And that is, what they did was they plotted moles of oxygen per flash of light per volume of cells as a function of chlorophyll in the system. They grew chlorella, the green alga, in many different amounts of chlorophyll, and then they plotted and they had a straight line. And this straight line was 1 oxygen but 2,400 chlorophyll molecules in the system. Why it was surprising, Otto Warburg, the Nobel laureate, had said at that time that you have every chlorophyll molecule that's chemistry. And if that was shown, then you should have been 1 molecule of chlorophyll per oxygen, but he had hundreds of molecules were used. So the concept of antenna was born in these experiments. And the experiment, so they wrote, we need only suppose that for every 2,480 molecules of chlorophyll, there is present in the cell 1 unit capable of producing 1 molecule of CO2 each time it's suitably activated by that. Of course, there's history to it. He didn't use the word oxygen, he used the word CO2, because Warburg had said that oxygen comes from CO2 and not from water. And Emerson was Warburg's PhD student. So you can see the language was affected, influenced by his professor. Here is Hans Gaffron, the discoverer of hydrogen production. That is, algae can produce hydrogen. Today we are going to exploit it. It's being exploited, how you can make hydrogen from algae and cyanobacteria. But he was the one who discovered that algae can produce hydrogen. He is the one, Hans Gaffron, yeah. I also knew him personally. He was in Chicago. Suggested that hundreds of molecules are together. There's only 1 energy trap, which he had called photoenzyme or sort of reaction center. It was 1936 paper. All right. So now we have this situation. You have an antenna molecule, and you have a reaction center. How does energy move? So first you have, let's say, 2 different pigments. Let us say you are a red algae. You are all red algae, and you have red pigment, phycoerythrin. And let's say you are chlorophyll. So if I give you a photon of light on you, and if I find the light coming out from here, what it means is that you have transferred the light to him. So that is the discovery of the excitation energy transfer from one pigment to the other. Something was already known in physics by Franck-Condon, James Franck. But in photosynthesis, it was studied by Lou Doizens in the Netherlands. His PhD thesis in 1952 showed excitation energy transfer from one pigment to the other in all systems possible. His PhD thesis is the classical thesis on energy transfer. However,
So the one second is one LHC complex.
This is basically a model.
Right. So when the light energy moves around the electricity, so they don't lose energy at all. I mean, it's
Very small amount of energy. You see, it is we're not counting the blue light, we're only counting the red light.
right.
The energy transfer is 100% from chlorophyll B to chlorophyll A.
No energy between the light types and complexes. You know,
No, these are within one. This particular slide
Oh.
is sort of represents only one complex where the reaction center is.
Oh, okay.
This is a model picture. It's not a real thing. Okay. So all it shows is a kind of model that any energy absorbed in chlorophyll B is transferred to the next chlorophyll with 100% efficiency, and among chlorophyll A molecules also transferred with 100% efficiency until it reaches the reaction center where the charge separation takes place.
But then, I mean, in practice, any movement will require certain energy, and 100 energy space.
It is a very strongly coupled system. So you have energy levels. The energy level is identical. Therefore, the loss of energy is almost zero. It's almost 99% of it. So the overall efficiency is 95%. But you lose, let's say if you are a red algae that you wear, then you would lose some energy. But if you were chlorophyll B to A, you will not lose energy. So depending on the system, you will lose. If the light harvesting complex was physically separated, you will lose energy. So you're right, you're thinking of real life. So that's good. Alright, so now let's go on. So this is a picture showing Steve Brody at the University of Illinois in Urbana. He's no more, he passed away. This is the picture when he was young. And what he showed, that when he put in light, so if you look at the experiment, this is a 1957 experiment published in Science. So he did an experiment in which he put light in phycoerythrin, and then he put light in chlorophyll directly. So when you put light in chlorophyll directly, you find the fluorescence going up rapidly within a few nanoseconds. But if you put light in phycoerythrin, he found then the chlorophyll light came after a few nanoseconds. So therefore, the energy was transferred, and he could measure the time of the transfer from one to the other. So that was the first such measurement. This shows you that everything is very dynamic. The system is not static ever. And your question is right, whether there is some energy loss. These energy losses are very small, and they usually could be heat loss. And there's a man named Franck-Condon in the Netherlands who has shown us that there is a really dynamic system available in this energy transfer. So this is a picture taken from Dr. Bruce, suggesting that everything is breathing, and all the molecules are not static, even though energy transfer is almost 100%.
Is it movement changing the light structure?
Yeah. So now this is a picture just showing you as a tool for teaching. You go to a garden. This is a garden in Heberkastel in Germany. And this is a picture taken by my friend, Professor Robert Clegg, who is no more. So that's why I dedicate my slide to him. He was a physicist in our university. And so photon comes in, E equals h nu, equals SCO lambda, and then goes to the reaction center very fast. Of course, I can show you going slowly, but very fast, and that's where the reaction takes place. Now, this is the slide, was shown just in August by Franck-Condon from the Netherlands. And Rink showed how an exciton can move in different systems. So he has an idea, and so he says the photon comes in, and the whole system is a little bit disordered. Even though it's disordered, it's very, very efficient in the system. So let me show you again, those of you who missed. So photon comes in and moves around, And this is a concept of what we discussed with some very coherence, as if it belongs to all the molecules. Where? Are you there? Yeah. So it belongs as if to all the molecules. But these are strongly coupled molecules, even though they are disordered. And then finally, just very quickly, again, show you that it reaches after, like a wave, and slowly it should go down. I don't know what happened. Maybe I have to click again. Something has happened. Yeah, there. Of course, it's like a football game. It's there. That's the goal. You reach the goal. You've done the chemistry. So, another way to represent it. As I say that when I teach, I prefer to show you slightly different ways. So one of them impresses you more than the other. So here is an antenna. We talked about the antenna. There's a large variety, as I said, and as you see, an exciton comes in, light photon comes in, it goes, excites molecule. Now here, I show you going 1 to 1. This is classical Forster theory system. And then the reaction center charge separation takes place, and the electron moves out. Now, the real system is cyanobacterial system. We have many molecules. You have phycoerythrin, phycocyanin, allophycocyanin, chlorophyll, and there's a cascade of energy. So when you go from one system to the other, it is Forster's theory. However, within the system, you could have what we call coherence, and you'll see the pictures of coherence. So now, before I go to the coherence, we look at the pictures of the master of the theory. The master of the theory is Theodor Forster of Germany. So here, what did he say? And he is teaching Bob Knox at University of Rochester, and they had the frisbee to show to students how excitation energy can move. And so what had Forster said? Forster said the energy transfer from one pigment to the other will depend on 1 over r to the 6, where r is the distance between the donor and the acceptor. And kappa squared, where kappa is the orientation factor. So if the molecules are dipoles, if they are like this, you're not going to have energy transfer. If they are parallel, you have energy transfer. And last is overlap. So if you have an energy level diagram, so if you have a lower energy diagram, lower energy molecule, let's say is chlorophyll B, chlorophyll A, the energy can go downhill very easily and lose some energy as heat, but still the quanta is 100% efficient for the quantum, not for the energy. And so this was a picture taken in US when Forster had visited Rochester. And this is just to show you how complex the system is. Merrick Senner and Doug Schultz's lab made this picture, and it's on his website. I'll take it. Now, coherent versus incoherent. This is the topic of the day. Everybody's saying, how? What's the mechanism? So what's the difference between coherent and incoherent energy transfer? Incoherent, there's a hopping of excitons from one to the other, which means the exciton means excited electron, which has a photon energy in it. It is localized. It is at one time it's one place. It's on you localize exciton, and then it hops. So that is the incoherent transfer. The coupling is weak in this case, or very weak or weak. And coherent is exciton is delocalized, like in the top picture. That's the exciton. It is all coupled, and you don't know where the exciton is. It basically belongs to all the molecules. And so this is delocalized, and it belongs to many molecules, and motion is somewhat wave-like. The coupling between molecules is strong. In light harvesting, this is Professor Greg Scholz, who had worked with Graham Fleming at University of California, Berkeley. And I have written a chapter in which we say that in light harvesting, we usually find predominantly incoherent energy transfer, but with some coherent character influencing the fast track energy. So in the steady state situation, you have to be careful of saying whether it's coherent or incoherent, not without further thinking or analysis. So this review will be in the next book, published in 2004. The title of our chapter is Photophysics of Photosynthetic Properties. All right. So who was the guy who really brought before us the idea of coherence energy transfer? Here he is, Graham Fleming. I was invited at the same conference to talk about incoherent and coherent energy transfer. So what he says is the first demonstration of coherent excitation energy transfer from Fleming's group at UC Berkeley and published in Nature in 2 papers. The quantum coherence allows reversible sampling of sight. In contrast to the hopping model, this wave-like motion helps avoid kinetic traps. Protein matrix plays an active role in energy transfer in the phenolmatrix Olsen protein that's used for green bacteria. Then also they wrote, like ripples in a pond, the wave-like motion of the quantum coherence allows the excited state to spread and explore many states. So it's a beautiful thing, but is it always so? Is it real? Always that we talk. But it is still a beautiful thing, and it's a part of the whole process of energy migration. So we have reached a place where you have reached the reaction center. So now you read the reaction center. Now excitation comes to you. What would you do to it? What you will do is, supposing you are the P680 molecule, of one of the molecules, and I am, let's say, the purified molecule, you receive the energy and you will give me your electron. Let's say this is your electron. So you have given me electron, you have a plus charge. So you have made a plus charge and a minus charge, and the charge separation is finished. And this we measured for the first time in Chicago at a laboratory of Mike Wasilewski. Here it is, Mike Wasilewski.
It's just enough to transfer the electron from the nucleus to the atom.
Well, the electrons are not in the nucleus. The electrons are already outside, all the outer electrons in the outer shell. Those are the electrons.
That energy makes the electron leaves.
The energy will make the charge separation, and then if something is there, picks up the electron. The purified is there. So these measurements were the first measurements in photosystem 2 done in 1989. This is what the time was challenged by Porter, and later everybody accepts. Now with the new measurements, we have new data. New data, I will show you. It comes from the laboratory in the Netherlands, Franck-Condon. All right, let's look at the new data. So this is a picture from Elizabeth Romero and Ring Franck-Condon. So it shows that once we understand this primary charge separation process, we could, in principle, do artificial photosynthesis and produce very efficiently energy. So you will have a division here who is interested in doing that. So it's very nice to see your institute where everything I'm saying is very pertinent there. So basically, as I pointed out, that you have the charge separation plus and minus. And now let us look at a little bit more details, again from Romero and from Mandela. So there is a P680 is the name of the reaction center of photosystem 2. And the two molecules, they're called PD1 and PD2. And there is another chlorophyll nearby, which is called chlorophyll D1 and a pheophytin. So what I showed, which I suggested, is that when you transfer to pheophytin, we were doing this charge separation. But Romero and Franck-Condon's group have found that there are two ways in which charge separation takes place in photosystem 2. So here, I will show you that result now. So these are the two ways, one starting in chlorophyll D1, another starting in PD1. So light comes in, and you have two types of charge separation, one between two chlorophyll molecules, P680 molecules, that are shown in the red, and one takes place And these are almost simultaneous events that take place because it depends on which photon reached where. And this is a discovery which is quite recent.
In our system, there is an energy transfer. Here there is charge.
Now it's a charge. Now it is a chemistry. So we,
always different.
well, the other systems, there is no partner which will accept an electron. So they're all equivalent systems. Now here we have the partners which have the redox potential and the chemistry, so that instead of exciton transfer, I'm not saying that they don't have before charge separation also exciton transfer. It's after exciton, because of the chemistry, because of the redox potential of the two molecules, you have electron goes back. Okay, everybody? Well, thank you for asking, because many people are shy to ask. So it's good. It's good you ask. All right. So now we have this charge separation. Let's see. You can see this one and this one, and then both possibility exists. So, what are the questions? I asked Franck-Condon, who is today one of the leading experts in this area. Of course, I don't like the fact that he believes in coherence versus incoherence, but that's okay. We can debate about it with him. So he wrote this for you. He said, how photosynthetic and other biological systems have learned to deal with their disordered energy landscape is an intriguing question. Because the entire molecules are not highly ordered. It's a bit disordered. There is clumsiness, there is motion, there's a dynamic thing. So he's asking, how can you do that? So we still have questions to answer. It's not yet finished. So let's see. Suppose we start with an excited state. Although there are zillions of pathways, but how and why they are not operational, but only the active productive one. And the efficiency is very high. So why are we interested in these things? If you are going to do physics and nanoscience, you better know these things before you can really succeed or understand these things. So here is the question that Link is asking, and he's saying we should all be asking to get some answers so we can build better systems. So how and why the light harvesting complex transfer excitation efficiently to reaction center complex instead of losing it as heat as it does during non-photochemical function. Link calls it controlled, he's given a new term, controlled disorder. Well, what does it mean? We don't know. We have to keep thinking. What is the physical mechanism behind it? We understand it all. We build better artificial systems than the nature has built for us. That is the point that he is making to us. Well, let's just go. Let's see, I don't know when I have to stop, but I will stop when I'm told to stop. But let me be a little faster now, because the energy transfer part is finished. Now we are leading you to the next lecture, which will be the evolution of the Z scheme that you all know about, hopefully. So here is the membrane. The process takes place, photon comes in here, photon comes in here, and we'll discuss it more. Protons go here, and they go to the ATP, make ATP, and NADP. For photosynthesis 1 makes reducing power, then it goes to the C3 cycle, as Donald likes to call it. He said the Calvin-Benson cycle, because it's a long term, Calvin-Benson, but it's okay, we call it C3 cycle. I'm not against it. Now, this is my detailed picture that is made by my associate, who I work with. I have no laboratory, so it's all by email exchanges and telephone calls. So we produce this scheme, the detailed scheme, and so you have photosystem 2, you have photosystem 1, the cytochrome B6 F complex. You may have talked with them about. I will skip any discussion of cytochrome B6F complex except to tell you it's very important, and it's important because there's a cycle, what is called the Q cycle, and extra protons come in, therefore extra ATP is made, and there is a cyclic electron flow from photosystem 1 to cytochrome B6 complex, and there are 3 pathways, 3 cyclic, so you have to recognize the fact it's not a simple cyclic system, one system only. So we will talk more about this scheme and how it was obtained. And I want to end this lecture by reminding you of what Buddha said 2,500 years ago. He said, believe nothing, merely because you have been told. Don't believe what I told you today. Just ask, read and ask and say, if this man is saying something that I don't want to believe, is he wrong? Maybe I am wrong, because I am a human being and I am guided by what I learn and what I read and what I think. And my thinking may be wrong. So therefore, he said, or because it is tradition, or because you yourself imagine it, this is the most important thing, that don't think you are faultless, and merely out of respect for the teacher. So also, this is very important Buddhist statement. That merely out of respect, but whatever, after due examination and analysis, you find to be conducive to the good, the benefit, the welfare of all, that doctrine believe and cling to and take it to the God. So the next lecture will talk about evolution of the disease scheme, and I thank you.